individual to individual. This one. Oh, sorry. So for today's outline.
Oh yeah, I'm sorry. They just started streaming. There was a little confusion outside, so they just started streaming. So you can go ahead.
Okay. So for today, I wanted to do three things, I think, for the talk. I'll give a brief introduction to a virus known as HIV, human immunodeficiency virus. And I'll give a little bit of background in terms of epigenetics, in terms of a more detailed understanding of potentially what that is and how it can be linked to viral infection. And then in the 2 second parts, I'll tell you a little bit about two projects, which I think are pretty exciting. The second project is a clinical project where we've been using cerebral spinal fluid from patients as a way to study a window into people's brain. For those that are interested in studying the brain, as we all know, we can do this post-mortem, but in living people, it's very difficult to access that compartment. But there is a unique ability to look within the cerebral spinal fluid that surrounds the brain and to interrogate the immune system in that compartment and to look at, in this particular project, viral infection in people living with HIV infection. But this approach applies to Alzheimer's disease and other things, but I'll show you some data from some new technology that can show you how we can study the epigenetics of immune cells in the CSF. And then part three, I felt like it would be nice to talk in a little bit about epigenetic aging. So how you could use these epigenetic biomarkers as predictors of brain health, but also other things related to the aging process. So I think just to start, if you go to look in the literature, and I think maybe for those that have been following, as we knew from the past 3 years with COVID infection, there seems to be this quite provocative link between viruses and brain dysfunction. I think the one that's apparently still controversial, but the one I think that received a lot of press is Alzheimer's disease, with potentially the link between herpes simplex virus and the outcome of the Alzheimer's. The one I think that now has pretty convincing epidemiological data is Epstein-Barr virus. So this is a virus that has been linked to multiple sclerosis. This was done in this very beautiful study in the US where they took samples of people that enrolled in the military and they did a health screening and they worked them up and took blood samples. But at the same time, they could look whether these individuals had or developed Epstein-Barr virus, and then later, 20, 30 years later, went on to develop multiple sclerosis. And with the epidemiological data, they were able to show quite convincingly that there was an increased statistical outcome of developing multiple sclerosis if you came in and you had Epstein-Barr virus. In terms of SARS-CoV-2, there's been a lot of data now with long COVID, COVID fog, COVID mind, really linking these respiratory viruses with viral syndromes. And then today I'll tell you about a retrovirus, human immunodeficiency virus 1. So this is a virus that predominantly infects people in their immune cells, so T cells in their blood, but also can get into the brain and infect the brain. The unique thing about this virus, HIV, comparable to the other viruses, this virus actually inserts its genetic material into our DNA. So those individuals in particular cells that are infected have their human DNA, but they acquire the virus DNA, and that's how the virus survives and replicates. I'll talk today about links between that virus and human-associated neurocognitive disorder and some of the issues we've been seeing, because with this virus, we have drugs that can shut off the virus. But if people stop taking the drugs, so say, for example, they decide, okay, this week I'm not going to take my antiretroviral medication, the virus will replicate quickly, and all the clinical symptoms will return very rapidly. Even though they're on viral suppression, they still have brain-related issues showing that the virus is still having a long-term effect. And I'll link some of those to neuroinflammation and some other things we've been studying. Just a brief background. So for those that aren't familiar with this virus, HIV infection, so this is kind of what the clinical trajectory or course looks like with an individual when they're infected. For those that maybe have an understanding from either movies or popular press, the real clinical outcome measure people use is a blood test for CD4 T cells. So you take a blood sample from a person and you measure the amount of T cells you have in your body. And these are cells in your immune system that are critical to fighting off flu, bacterial infections. These are kind of the fighter cells in your immune system that remember a pathogen you saw. So when you get vaccinated, T cells are the ones that remember and are able to fight off infection. When they get infected with HIV, the prominent syndrome in terms of clinical is CD4 count loss. So if you see here in blue, over weeks of infection, they get a decrease in CD4 T cells. And in red here, I'm showing you the amount of virus in their plasma, so in the blood. So as the T cells decrease, the virus increases significantly. And then over time, in about a year, people seem to reach this point where their immune system kind of balances out with the virus. But if they don't get treatment, what ends up happening is these people progress to AIDS and eventual death. So this is a process where you've probably seen in the popular press, watching movies on HIV infection, predominantly, I think initially in New York and San Francisco, but globally, virus that really at this point has no cure, but there is a treatment. But if you left it untreated, this was kind of the course immunologically, clinically, what you would see. In terms of just some demographic data, it's estimated around 38 million people globally are living with HIV infection. As I said, there's no vaccine or cure. The one thing we do have are quite remarkable antiretroviral drugs, similar for anyone maybe received if you were infected with SARS-CoV-2, like Paxlovid, something to reduce viral load. That's really been the game changer for HIV infection. And what I'm showing you here is a representation of what it looks like in an individual. So this would be their immune cells, high viral loads, like I said, lots of virus. If you don't treat, if you use antiretroviral therapy, which are drugs that shut off the virus and stop it from replicating and infecting other cells, you see essentially the body's able to clear the plasma. So clinically, if a patient comes into the clinic, they do a blood test, and then they measure how much virus is there. And if they're undetectable, they're untransmissible. So this is where people, as long as you're taking medication, you're not able to infect another person. And you can live essentially a normal lifespan, you won't develop AIDS. But if these individuals stop treatment, the virus comes back very quickly because, like I said, it's integrated into the DNA. And if the viral medication stops, you get this kind of rebound. The one, sorry. Avoid it.
That's perfect.
Yeah. The one thing I think that we should understand is that we're able to clinically test when someone can receive treatment based on an antibody test and a viral RNA test. So I think we should all be pretty familiar with COVID, right? The first thing you would see at the very early stages, if you do a PCR test, you get viral RNA positivity. And then subsequently, as the immune system sees the virus and learns, you get an antibody test. So this is our, when everyone was swabbing your nose and getting a COVID test, this is your antigen test. So this is in the clinic how we diagnose HIV infection. In comparison to COVID, it's very difficult to get people at this early acute phase. Typically, people don't present until the chronic phase, until they're very sick. They go into the clinic, people ask about their lifestyle behaviors, potentially, run blood test diagnostic, we do a test, and typically this is where it's diagnosed that an individual is infected with HIV, and then they start antiretroviral therapy. But as you can see, in terms of the course of how the immune system's impacted, this occurs quite early. In terms of the drugs we have right now, there's a myriad of drugs that are available. Antiretroviral therapy globally has led to an expected survival and increase in lifespan of 20 years. So people now can live into their 80s, 90s. The one thing, though, that is the issue is even though these people are living long lives, the quality of life seems to be different in the sense that they have a lot of comorbidities and brain-related abnormalities due to potentially residual inflammation that's in the brain. When we look in the brain, particularly in CNS, the virus gets in very early. So there's been beautiful work where they've looked in CSF samples of people that they can capture within a week of infection, and they can find viral RNA in the CSF. So the virus gets into the CNS early. There are instances where some people, even though they have antiretroviral therapy, for whatever reason, the antiretroviral therapy doesn't get into the brain because of some of the toxicology related things in terms of clearance. So you still get active viral replication in the central nervous system, even though in the peripheral, clinically they come in and it's like, okay, they're undetectable, untransmissible. But in the brain, there seems to be some residual RNA, suggesting that there's like this compartmentalization of the virus that may be occurring. In terms of autopsy studies, a lot of elegant work, depending on where the virus infects, it likes microglia cells and T cells in the brain. You can detect with PCR and qPCR, HIV, DNA, and RNA in around 50% of the tissues people look at, but you can imagine the brain's quite large. So it's really kind of an idea of finding a needle in a haystack, but there is evidence that the virus is there and it's invading. And then, like I said, there seems to be compartmentalization. So if you look at viral sequences between the blood and the brain, the virus seems to be adapting to the brain in a different way because the immune system is a little bit different in the CNS. So I think just to summarize briefly, the two instances you would have, if you have access to care, You have access to medication, which I think, in terms of globally, we've done a remarkable job of rolling out antiretroviral therapy testing. You have an instance where you have people living with HIV on suppressive ART. So ART being antiretroviral therapy. And when you look at the neuro impact, even though this happens, the reports, depending on where you are in the world, around 19 to 69% of people report having a cognitive issue that seems to be related potentially to HIV infection. And this cognitive abnormality seems to occur with any CD4 count. So you can imagine, right? You would think, okay, if you have the lowest CD4 counts, maybe then your brain would be the most impacted. Not necessarily. We even have people that have high CD4 counts, which is kind of a measure of immune health. Those people still show these cognitive abnormalities. In terms of the clinical measures on CSF biomarkers, it looks clinically on ART, everything looks quite normal. So it looks like, there's no provocative kind of Alzheimer's markers lighting up or inflammatory markers. So there seems to be something that we're not able to measure completely that seems to be residual. And then in terms of imaging findings, it's been variable in this instance. In comparison, when we didn't have antiretroviral therapies, there were quite provocative brain-related abnormalities related to dementia and AIDS. You just let the virus replicate without any control. The impact on the CNS was quite dramatic in terms of what you would see. And this was referred to as HIV-associated dementia, which currently in the current treatment era, we don't see people with dementia per se due to HIV. It's this mild cognitive impairment that really is impacting daily life. Why we think this is occurring? We think that a lot of factors are playing a role. The one that we're particularly interested in studying is neuroinflammation. But you have other things that are related to comorbidities, so high rates of diabetes, obesity, other things related to trauma, violence, stigma, environmental factors in this population. You also have other things related to drug use. So if you're taking antiretroviral medications, they could interact with other drugs you're taking. This could lead to other things that are cognitively creating issues. And then there is this, I think, ongoing idea that you have residual virus replication. So the virus is residually replicating and causing a little bit of residual inflammation that's leading to these issues. So when we study it in the lab, we're really interested, I think, in two different arenas. That would be, I think, the most relevant when you're looking at the CNS. Those would be brain autopsy studies and cerebral spinal fluid studies. This is, taken from all the neuroscience field, right? If you want to study people, either look at brain pathology with postmortem brain tissue, or you look at living people like a lot of neurology groups have done by accessing CSF. And in the CSF, remarkably, you can look at some immune cells, which I'll tell you about. And these cells can be interrogated for molecular molecules, so like HIV, RNA, and DNA, but also post-HIV or post-DNA and RNA. We also can look at the proteins in this fluid, so you can look at biomarkers that are individual in terms of things related to neuronal injury, such as neopterin or soluble cytokines. These are molecules that the immune system releases as signaling them. And then in terms of looking at neuronal glial loss, you also could do some imaging studies if you wanted to as well. But those aren't studies that we currently are doing ourselves. So for my lab, I've really been interested in this homeostasis between the brain and the immune system. So we're really interested in studying the immune cells, both in the periphery, the CSF, and in the tissue that are related to a myriad of things that are impacting microglial function, astrocyte function, and then also potentially neuronal signaling. So this is kind of the framework for what we've been looking at. In terms of how the virus can impact the brain, and 3 potential mechanisms are occurring. The one that I've been studying is the immune response in terms of infection. But the 2 that I think are those that are early entry kind of mechanisms, if you wanted prevention or to look at the earliest stage, would be direct crossing. So this would be virus from the periphery, because these people are acquiring HIV not in their brain, right? It's not like something's going on and they're getting injected into the brain. So it's coming from the periphery in the blood into the CNS through this blood-brain barrier. So this is one potential route of how the virus gets in. The other is this Trojan horse idea. So this is the idea and analogy that the virus hides in a cell, and these cells naturally are going back and forth between the brain. This is the one I think that has the most convincing evidence how the virus gets in, so this Trojan horse. And then once that happens, cells throughout the immune system can be recognized that, hey, something's not right here, there's a viral infection, and those cells change. And this is the process that we're interested in studying in terms of how these cells change in regards to epigenetics, because it's not, for the most part, overwhelmingly, like every cell in the brain's getting HIV inserted. the majority of 99% of the cells don't actually have the virus, but their function changes because they can recognize that one of the cells is infected. And this is really where the process we've been studying. So we've been looking at epigenetics. The best analogy I can give is your DNA, you can think of as a musical score. The conductor is essentially the epigenome, and it's notating this DNA or musical score and playing a piece of music in a unique way. So all of us would essentially have, for the most part, the same genetic code with some variants. But if you look at the manifestation of all of our appearance, cell function, age, this process, your DNA is not changing once you're born. But what is changing is the tuning of kind of this music, which is your DNA, by epigenetic marks written in pencil. So pencil is kind of the analogy. These things can be erased, they can be put back down. So this is essentially what it is. The markings you can think of are your DNA code, and the epigenome is the pencil erasing. The other analogy which I like to give is if we're looking at which genes are turned on and off, this is similar to looking at a stop sign or a green light or red light in terms of studying which or telling which genes to be on and which genes are to be off. In terms of HIV, on this unique way to study this interaction between the host and the environment. So you have a virus which is interested in hijacking or taking over a host. And similarly, you have a host which is interested in regulating the virus. So these epigenetic processes are occurring between the environment, which is the virus, and the host, which is our immune system. And we're looking at what the virus is doing and what the host is doing to try to overcome that. But you can imagine this could be applied to anything. This could be toxic exposure, this could be stress, this could be violence. It's kind of this interplay between these two things that is leading to a dynamic process that is associated with disease. In terms of this context, I'll be telling you about CNS disease. So for CFF studies, that aren't familiar, this is typically performed in the clinic by a neurologist. A lumbar puncture, a needle is placed into the compartment where they're able to withdraw CSF fluid. And then this fluid, typically, if you spin it down, you can get cells. Most people, if we look at most of us, and let's say we take 15 milliliters of CSF out and spin it down, we have about 1,000 cells. Very, very small amounts. So a pathologist typically would not say there are cells because they look under a microscope. They're looking for leukocytosis, which is essentially the buildup of lots of immune cells from the CNS related abnormality, where you get like hundreds and thousands of cells that are immune cells in CSF. Most people have around 1,000 or so, but it varies among individuals. I've seen people that are normal, completely healthy. They have 50,000 immune cells. I think these factors are dynamic in terms of when things are trafficking through the CSF. The CSF compartment gets into the ventricles in the brain, and it, for the most part, in classic neuroscience, is thought of as the cushion for the brain, but also is the vehicle with which things are brought in and out of the brain as a waste clearance mechanism. And there's a lot of new data showing this lymphatic system that is a waste clearance mechanism out of the CNS. But there are immune cells in there, right? So clearly there are cells that are functioning within this compartment for a reason. In terms of what you can do with these fluids, for CSF, just the fluid itself, this is clinically used to look at cytokines, antibodies, markers of blood-brain disruption, markers of neural injury, ATN biomarkers. If you can get access to the cells, we can look at the epigenetic signal in these cells, so how these cells are acting in terms of on or off, or what kind of cells are even there. To do this, we've been using a technology, so it's a couple thousand cells, right? We couldn't be essentially isolating large amounts of blood and purifying them. So what we do is we use a fancy microfluidic technology that puts the immune cell in this oil droplet. So we're able to do reactions to look at, when we look at the epigenome with sequencing, each individual cell. And then we can treat these cells in a plot in terms of how they look in relationship to other cells or other people, which we're able to look at the function of these immune cells in their epigenetic landscape. To do that, I've been working with the team. This project is, I think, 3 years old now. These are investigators at Yale in the neurology department and computational biology, where we recruited people living with HIV on antiretroviral therapy. We also recruited healthy controls. We had an individual living with HIV before they got treated longitudinally, 3 months, 7 months, 9 months. So this individual had high levels of virus. We were able to get CSF samples and blood, and then follow them after they went on treatment to see what happened after treatment. Like I said, we took clinical specimens of blood and CSF, and then we looked at both the epigenetics and the virus in these cells. So what I'm showing you here is combined blood and CSF. As you could imagine, there's cells we have in our blood that aren't necessarily in our CSF, but we also have cells that are shared between the CSF and blood. These dots here are each one individual immune cell, and we're able to look at these cells and identify what they are. One of the major things we've seen early on that was quite striking to us was that there were quite a few cells that were infected within the CSF compared to the blood. And when we looked at these cells that had virus in them, so this was HIV virus, these cells were the ones that were producing high levels of interferon. So this was essentially telling the body and the immune system that, hey, the virus is still here. And this is potentially, I think related to the immune system not being shut off, even though these individuals had antiretroviral therapy. So we're able to see really it was interferon signaling that was a major driver of activation in these individuals. The other thing we saw, which was really interesting, so in your blood, you have T cells, B cells, monocyte cells, and other immune cell types such as NK cells. Those are all shown here in these plots. But the fascinating thing, only in your CSF, you have these cells that look like they're microglia cells. They're not fully developed microglia, but they don't look like other cells that we find in blood. So it suggests potentially that there's a unique immune cell subset that's going throughout the CSF compartment that could be a driver of inflammation. So what we decided to do was focus on these cells. And when we looked to see kind of epigenetically what was on in these cells, we saw some very interesting things. They were all enriched in genes that had been identified in neurodegeneration, so such as CD204, which is a scavenger receptor, TREM2, which is another receptor expressed on monocyte cells, ApoE, which is a known neurodegenerative gene process, and also other genes related to caspase D. We took the cells and you could ask, okay, which cells are interacting with other cells by some fancy bioinformatic methods, which ask the question of, if this cell has this receptor and this cell has this ligand, which cells essentially can go together? We were really struck in the CSF, just among those 2,000 or 5,000 cells, there's a lot of interactions between these immune cells. So if one cell has a signal, there's a virus, there's an infection, it's telling the other cells that a lot of things going on. So we're pretty well convinced that it seems to be a perturbation potentially in a cell population that's leading to dysregulation in the other cells. So it's this intercommunication that seems to be the driver. And when we look at the microglia cells, this is a plot just showing you, okay, this is the CD204-like cells I told you in the CSF. Which cells are they putting messages out to? And we looked at particular genes that they're really driving. The one big one that popped up was complement signaling. So for those that aren't aware of complement signaling, it's really been a major driver of an inflammation related to neurodegeneration. And recently has also been shown, in terms of this long COVID work, a signal that really is driving perturbations in the CNS related to persistent neuroinflammation, which would affect CNS behavior. We also found differences in osteopontin, and osteopontin is interesting because it's a molecular break. So this is a molecule in the CSF and in the tissue that would shut off. So for most of us, if we get a viral infection, so let's just say you get infected with influenza, the flu, you go through the course of influenza, you probably don't feel great. Your brain probably feels a little bit off, foggy. Then you resolve. Well, really what that is the immune system shuts down after a point. Once it knows, hey, everything's fine, I cleared the virus, shut off signal. Osteopontin in the CNS at least is doing this. So it's shutting off all of these pro-inflammatory signals, but there's been data shown in plasma and other samples people have access. This protein is still elevated, so this signal doesn't necessarily shut off in certain indications. But in these CD204 cells, we find that they're a major outputter to tell, hey, let's dysregulate this pathway. So we think that this is a way that the system is dysregulated and persistently not shut off. We followed up on these studies and had a unique opportunity to move beyond CSF and to look without the body. I think for those that are in the neuroscience field that are starting off, the field is really, I think, rapidly getting into the idea that the mind is connected to out the body. Typically, it was the gut-brain axis, but there's evidence there's a lung-brain axis. Throughout the whole body, there's a communication aspect. So the CNS is getting a sense of what's going on in the periphery, and the peripheral organ systems also are communicating to the CNS. So what we decided to do was access the CSF in an individual before infection, but also look at their blood, their gut, and then also in the lymphatic system. So these were clinical samples where we're kind of cross-comparing across all the compartments. And this is just showing you how each of those compartments, each of these is just a cell, but the take-home message is each of them is made up of different cells. And we know, right? If you look in your brain, those cell types are quite different than the cell types in your gut. Those cell types are quite different than in the lymph node. But the compelling part that we found, when you look at the virus is everywhere. So it makes sense when you start to look at this connection between these outcomes and comorbidities. These people have cardiovascular disease, they have gut perturbations, CNS perturbations. The signal from the virus is getting interconnected throughout everything. And if you have residual virus here, it potentially is interacting in the blood is sending signals potentially to the gut potentially is interacting with the lymph node. So there's this cross communication that's occurring throughout the whole body. So while we like to typically focus in one compartment, we have to consider that everything is really communicating. What happens when you shut off antiretroviral therapy? So these are just some numbers before treatment. As you would imagine, there's a lot more cells that are infected or have virus. After treatment, the interesting part is we still can detect these cells. So there seems to be this low level. Clinically, these aren't meaningful at all because they're not clinically detectable on a PCR test. But at this high resolution, it's telling us that the treatment and medication isn't shutting off things completely. So this is really, we think, the issue. So there needs to be some effort to, I think, somehow continue to shut these cells off with better drugs, or even efforts by gene editing to turn them on. So I'll switch now, I think, to the second part, which is, okay, I told you some data on studying immune cells and looking at their signals in terms of which genes are on and off. The other thing we can study is the marks that are on the DNA. So this is kind of that pencil marking that is occurring on your DNA code. Typically referred to in the epigenetic field as DNA methylation. So this is a covalent modification. So it's a chemical change that's on the DNA code. In the lab, we can get DNA samples and measure this. We've been able to use this data to predict biological age. What groups have done is they've taken data from, let's say we have 10,000 people from birth until they're 120. And we have DNA samples from every single person. We take their DNA and we measure their DNA methylation marks. And then we know their chronological age because we have data from when their birth date is. And then we train models to identify which of those methylation marks are associated with aging chronologically. So that's been kind of this epigenetic clock that's been built from these initial first clocks in 2013. What was found, I think, in the first clock from Steve Horvath's group, when you do this throughout the whole body with like 99% predictive accuracy, 353 parts in our genes could be measured in terms of methylation, and then you can take any DNA sample from any person, not know their age, and you could reliably predict what their age was just based on this mark. So you can imagine this is quite interesting in terms of, well, what if your age is a lot younger biologically than it is chronologically? Or what if it's higher biologically than it is chronologically? So there's been a lot of work that has looked at that difference in terms of age acceleration. That's been associated with every known disease outcome you want to look at. So they went back and retrained these models and have made these mortality predictors. So let's take people 10,000 that we have this data on, we know their chronological age. Well, let's follow them and let's predict when they die. And then let's use that data in our model and retrain it. And then apply that to other people. And what you could do is basically say, okay, I have a DNA sample of me today. I look to be this age based on this chronological age predictor, but the mortality predictor is elevated. So it suggests that potentially, I'm biologically older in terms of mortality risk. And when they look at people that have diabetes, their grim age, in reference to the Grim Reaper, is older, so their mortality risk is increased. So it's a very good mortality predictor. People that are also having cancer are elevated, and as you can imagine, people living with HIV are accelerated. So they're closer to mortality than people that don't have the virus, and we think it's potentially related to this residual inflammation and other systemic comorbidities that are occurring. This is just a nice summary in terms of a graphic, very simply to explain what I just described. Chronological age along the x-axis, epigenetic age or biological age along the y-axis. Most people fall along this solid line, which would be kind of a linear relationship. But if you look at each individual, depending on their comorbidities, other variables, they can deviate to be higher on the line or lower on the line. People that are these extreme agers, so centenarians, they appear to be a lot younger epigenetically. Makes sense. They live to be 110, 120. You have other people that have age-related diseases that they die earlier. Their epigenetic age is significantly increased. So this has been kind of a good biomarker. And the aging field has taken this recently and applied this to a variety of different contexts as a way for clinical interventions to see whether if we change something, your diet, exercise, can you shift your age in one direction or not? I still think we still need a little bit of data to build better clocks, because you could imagine there's not a lot of data throughout the globe. So, predominantly a lot of this data has come from the US. If you apply those data sets in some other populations, they don't necessarily match perfectly. So, it's an exciting area, I think, in terms of additional epigenetic data that we have. And then this just summarizes kind of what I've been talking about in terms of epigenetic clocks and a myriad of things that are environmentally related and health outcomes. I think the most exciting one is probably the behavioral interventions in terms of diet and exercise. We all wish we could take a pill and magically have something get better. But for the most part, it's the hard work in terms of diet and exercise seems to be the biggest stimulus. In terms of other things, cancer, these are things we know that are accelerating epigenetic perturbation. And then also, I think social determinants of health, infections, like I told you about, and also things related to neuropsychiatric disorders are all impacting, this measure biologically. So I'll show you kind of how you can apply this to summarize in the data I showed recently. So you have an instance that I talked about. This is an untreated viral infection. This is an individual that goes in antiretroviral therapy. And then this is just a healthy control from the same population. And then we took blood, we looked at the methylation patterns, and then we looked at some epigenetic clocks, and then just asked the question, okay, if you have treatment, what does this do in the different immune cell types in the body? What I'm showing you here to start is throughout your body, if you isolate different cells or organs, your body has a different kind of age, right? So we could compare whole blood among everyone and get a measure, but you also could just look at T cells, or you could look at monocyte cells. So in this instance, if you just pick people that are healthy control, the monocyte cells appear to be older than their T cells. So the T cells are about 8 years or so, 6, younger than the person's chronological age. If you look at people living with HIV infection, before antiretroviral therapy, their T cells are about 9 years older than their chronological age. Makes sense. Their immune system not healthy. It's going to be related to potentially, if they don't get treatment, AIDS and then eventual death. If you give treatment, significantly decreases, makes sense. You decrease the virus, you increase lifespan, you increase the health of the immune system, but it's still not at the level of what you would see in a control. So the 0 would be kind of a good match, for most of us, we wish we were a lot biologically younger than we are chronologically, but you hope to be somewhere around the 0 time point. Surprisingly, when you look at these other cells, these monocyte cells, so these were the cells that are kind of being pumped out of your bone marrow regularly, and they're trafficking in the brain. They're also the cells that are related more like to the microglia cells that people are interested in studying. These are cells that are inflammatory producers. After antiretroviral therapy, these cells don't get younger. They seem to even get older. So we think that this residual replication seems to be increasing kind of biological aging in those cells. And then the other plots are just breaking them down compared to controls. So these are the controls. In this population, when we looked at 100 people, they're about 5 years younger than they should be, which is good in this population. This is people living with infection, like I said. They decrease, which is good with treatment, but they don't ever look like a control, so they still have this difference. And then these monocyte cells are younger in controls, but even with treatment, they get older. So, there's really this dysregulation that's occurring with people living with HIV that, in terms of biological aging, there's really a scar there that's not being resolved by treatment, even though, it's overwhelmingly incredible that they can live a long life, not develop AIDS. But in terms of things going out throughout the body, even in the brain, there's a signal there that's been hijacked. So you can take this data and then longitudinally follow people. We've been able to kind of do that 2 years after they've gone treatment and look outcomes. I'll just show you some data in terms of some clinical outcomes. The more clinical data you have when you do this, the better. But for us in this context, we were really interested in a couple things. No serious clinical events. So for example, you get treatment, these people shouldn't develop AIDS, they shouldn't die. That's one thing. No grade 4 adverse events. Their viral load should be low on these clinical blood tests. Their CD4 T cells should be recovered right because they're getting treated. their CD4 to ratio, which is another measure of health, could be greater than 1. And then we did some cognitive tests, cognitive impairment by MTZ score. So we said, okay, they needed to have no cognitive impairment. And we just looked at a biomarker of inflammation. And we just were asking the question, okay, could the epigenetics kind of predict out what the clinical phenotype in some people were? Because some people seem to improve or stay normal, and others seem to decrease in some of these clinical variables. In terms of some of the data the group has shown, is just a take-home message in terms of cognitive data. Each of these lines is kind of tracking cognition over time. You would think, right, homogeneous population, everyone with the same treatment, there's individual variation among everyone, so you have all the different trajectories. But in terms of clinically, you have to wait 2 years to know where someone gets to. We just were asking, okay, can we predict out what may be going on? So what we did is we took all the epigenetic data, and then we looked at outcomes with some clinical data, right? So we're taking like their T cell counts, their age, their viral load, and then the monocyte epigenetic methylation data and their T cell epigenetics. And then we just looked at the outcome of these 1,000 people, whether they had an unfavorable outcome or a favorable outcome clinically. And then we also looked at whether their neurocognition increased or decreased over time, just to kind of see, okay, is this a good additional biomarker to clinically what the clinicians are following? Because they typically look at CD4 count, and CD4 count is kind of the measure of like, oh, how good's your immune health? And what we found interesting was that the CD4 count still was a feature that was quite important, predicting longitudinally. So if you had recovery of your immune system, that's great, and that relates to neurocognitive and clinical outcomes in terms of favorable outcomes. But it was interacting with the methylation marks that were from those inflammatory cells. So the inflammatory cells that were epigenetically worse, those seem to be related to worse clinical outcomes versus those that seem to be better in terms of the inflammatory immune cells epigenetically in terms of methylation. If those seem to be better or normal, clinically, it looked like those were a little bit better in terms of the clinical outcome. In terms of our efficacy, we're now on the current process of replicating this in another cohort. This was done in collaboration with the cohort in Asia. So whether this applies in the US, South America, it's unclear in terms of an out of sample validation, but at least for us, it gave us some interesting insight that the epigenetics is an additional kind of biomarker that we should be measuring in terms of when we look at long-term clinical outcomes in these longitudinal studies, because it can change. The DNA will never change in terms of the sequence, but the epigenetic profile would change over time. So I think just to summarize today, I hope I convinced you that it's worthwhile to look at links between viruses and brain dysfunction. This is a very interesting area in terms of you get infected with a virus, what's the long-term health outcomes of that, but also too, throughout your life, the pathogen load in terms of different viruses you're exposed to throughout time. We think this has some relationship to eventual brain abnormality and dysfunction in some people. I gave you an example of the retrovirus HIV infection and showed you that it has a long-lasting impact on the CNS despite antiretroviral therapy. So even though you can treat this virus, seems like there's a residual effect that's there. For those students that I think are interested in doing translational work, I showed you the kind of magic that can happen with CSF biofluids in terms of trying to study some epigenetic or molecules in the CSF in relationship to interesting things. In this instance, I showed you in terms of viral infection with HIV, but I think CSF biofluids outside even of the context of neurodegeneration is a very interesting thing to study. There's not many doing CSF studies, so I think it's a worthwhile thing to spend time learning and trying to get partnership with clinical neurologists and others that are doing lumbar punctures in a research setting. And then I showed you some interesting data on these interesting cells in CSF that we've identified. We're calling them CD204 microglia-like cells. So these are just cells that we think are kind of the drivers potentially of immune dysfunction in relationship to infection, but also in regard to potentially to neurodegeneration in terms of kind of continuously telling the immune system and the brain that, hey, something's not right here, let's continue to activate. And then lastly, I kind of hopefully showed you some data on this emerging field of epigenetic aging and epigenetic clocks, and how you can use some of the epigenetic data with some informatic methods to do some predictive modeling, whether that's chronological age, but you can imagine you take all these methylation marks, you could do machine learning or lasso regression with other data sets and predict out other biomarkers. So let's say you have data on whether someone has a certain cognitive behavioral profile, you can make a risk profile, and whether someone has this certain profile, cognitive behaviorally, or this profile, you have enough data based on the methylation marks. So you can say, okay, it looks like potentially we might have this person with this particular molecular phenotype that matches the clinical phenotype. So this is kind of a powerful approach with the data in terms of data science. Yeah, just adding acknowledgements, have been lucky to work with a lot of different groups across the US military, globally, through industry, and have been supported by the NIH for a variety of different institutions. But yeah, I'd be happy to take any questions.
So thank you so much. We have time for some questions. If someone.
I mean, I have one
jump.
question. I first I need to say that I'm not a doctor and I do not come from the neuroscience field, like in my background. So I don't know if this is a relevant question or not.
Any questions though?
But when you were mentioning about the different types in which we know that the cells are like the HIV cells are in the brain, you mentioned the second type. And you said 99 cells do not have the virus, but there's 1% that do. How does that happen? How does they know, like, how do they know that there's 1% that do? Do the other cells know? I don't know how to put this in.
Yeah. So we have basically a cell that has a door, right? That the virus likes to go in. So when the virus replicates, the way the virus finds the way to survive is it goes in that cell as a home through a certain door. But if we look at all the different brain cells, all the immune cells, only a certain proportion of cells have that door.
Okay.
So it's like a magic door that only the virus can get in.
Oh,
So it uses that receptor.
different cells. So same cell, but different doors.
But within that family of, let's say, okay, every cell has that same door. So you have, let's say, 1,000 cells and they all have the same door. It's just a matter of the immune system and the virus being able to match up together. So what ends up happening is this rarity thing where the cell gets infected, the DNA is inserted into that DNA in the cell, and then the virus is tricky. It says, I'm going to hide. So it shuts off, and then it only stays in that cell, and then it'll replicate, produce more virus. And then the cells that essentially become infected, because it's not good to have kind of like this invader, those cells will die. So what ends up happening, that's why those T cells are tanking. You get all these cells are dying. But when you treat someone, the treatment is essentially shutting off that door so it doesn't let it locks the door, or it's on the DNA stopping protein production or viral replication at certain parts. So then you have kind of this instance where it's like a needle in a haystack. But if you stop treatment, the virus just randomly says, okay, I need to make more copies. And then it just continuously comes back and increases. So that's been the issue with the vaccines. Every vaccine, if you want to talk about money wasted, not wasted in the sense of what was leveraged for COVID, but billions and billions and billions of dollars, the smartest people in the world, every vaccine has failed, miserably failed. They vaccinated, you get infected, it doesn't do it. The only thing that they've been trying to shift to now is to try to get the virus to come out of its hiding, but that's also challenging because the virus is very tricky. It's figured out a way to evade the immune system and evade basically the killing of these 1 in a million, but somehow the body is not right. It knows something's hiding here. So that's kind of this thing where these cells are producing molecules which are telling other cells there's an invader. So a signal's going off, but for whatever reason, we don't have the ability to shut off that signal.
Yeah,
so,
we don't exactly know where it's coming from, but we know it's happening.
Exactly.
So we don't, okay.
So that's, I think the surprising thing for us when we did this, at least clinically, to start, was we were surprised that in people that are walking around that are undetectable by a clinical test, when they go in the lab, they're not going to infect anyone else. They're clinically undetectable. If you use very high resolution tools, we can still detect virus replication, very low, but it's still there. So it's telling us that there's this rarity thing, but also this thing of the notion that everything is shut off completely. That's also not true. But the thing is, the message, I think in terms of clinically, in terms of the press, the public, clinically, it's undetectable by PCR, but very, very high resolution, there's dysfunction still occurring. But yeah, great question, because that's kind of the interesting with HIV. It's like, it's a 1 in a million question. And then every person is also interesting. So if you look among different people that have the virus, some people have so low amount, and some people have a lot higher amount. So their starting points in terms of how good their immune system is to kind of kill the virus or regulate it. It's this kind of balance act that occurs.
It's very interesting. Thank you.
Yeah.
Mike, I have a question regarding to in terms of framework. as you and me want to do regarding to the red lab, which is like to try to capture any epigenetics and try to assess potential links between those markers and I don't know, brain health measure, which is brain health measure could be considered like a big measures. So you are presenting here subtle changes in the number of, I don't know, monocytes and CD4, CD200, 204 cells. So I was wondering what kind of link you could track between the subtle changes in those kind of inflammatory measures, inflammatory cells, and how you could impact like a bigger measures like brain health. I mean, I was wondering in terms of, you know, you could use PET in order to track, I don't know, beta amyloid or p-tau or tau imaging, but also you have, you know, we have option here to assess like a atrophy, brain volumes, cortical thickness, but those kind of measures appears to be so bigger. I mean, in terms of just the kind of results that you are showing up. Yeah.
Yes, I mean, I think depending on the compartment you're accessing. So
Yeah.
if you combine all the blood cells, there's a signal there still in all the blood cells that are related. But the way to think of it, I think, is so you have all the DNA. There's 23 million pencil marks.
Yeah.
300 of those marks so far have been related to chronological age. There's other marks related to brain imaging findings. There's other marks related to smoking. So there's like different pencil marks that people are identifying. So depending on what the outcome measure is in terms of the analysis, that's where the link comes. And then when we look at it, there are things that are related among different things, right? So there's marks that are related to cancer that appear to also appear in aging that also appear here. But interestingly, I'd say that's less than 10%. That seems to be a common mark between all. So the immune system, when it's dysregulated in terms of these outcome measures, while they're direct or not, that's unclear. But the biomarker, it looks like the signal is pretty stable. At least for the imaging stuff, I was still struck by, you can take a blood sample and predict what someone's hippocampus size is.
Yes.
So that to me is like, it doesn't make any sense, but then it kind of makes sense if the immune system knows that there's hippocampal degeneration occurring. It's picking up on that because the immune system potentially is more inflammatory. It knows that there's neuronal death. So there's a signal that's kind of changing things and stably at a point that we're able to detect in people that have, you know, normal, bigger hippocampal sizes. And that also relates to some of the aging marks. So yeah, I think as we get more data, it's an exciting area, but for neuroscience, you need the matched data sets to do that. And that's where the people that have, like ADME has imaging data, biomarker data, clinical data, they have epigenetic data, you can make those connections. So that's the exciting part in terms of working across discipline. And then the next step is to show that it's causal. You know where those marks are. We can now epigenetically edit with some fancy CRISPR technology that's been used to cut genes. So you can go and turn on these sites at specific regions and then look in like a mouse model or in vitro. Does this change memory? Does this change, you know, things that are more linked directly? And I think that's the direction people are moving, is the associations are a start and they guide you like what you should be doing in terms of mechanism. But yeah, you're correct in that. I think at this point, we need more data from these high level neuroscience kind of brain health measures to link out, you know, what the mechanisms may be. But I think clinically, it might just be enough to say, this person has a high risk of having low brain health in the next 3 years. So then you might be able to try to say, okay, let's maybe give them some positive clinical, you know, interaction to try to promote healthy behaviors and other things to try to stem off that risk. I guess it's similar to ApoE.
Yeah.
In terms of, you know, you test ApoE.
If you have biomarkers, you could introduce those marks as like a mediator, so the model between methylation and brain health measures. I mean, it's just introduce multiple levels of analysis and try to see potential interaction between each other.
Yeah, but I think the methylation started as like the best biomarker, one of the ones they were really excited about for cancer.
Yeah.
And it's revolutionized the cancer field. So you can go into a clinic right now, give a blood sample, and they look at circulating DNA and look at the methylation patterns, and 99% predictability, they know what kind of cancer you have. Just based on this liquid biopsy.
Yeah.
So they don't have to go through a biopsy. They just do this liquid biopsy. So it's worked in that regard. So for the like brain health, you can imagine if you go into CSF and do epigenetics of methylation of proteins, you can say, hey, the neurons are all dying, so we're picking up on it. So that's, I think, where the field is moving. But, neuroscience and immunology, I think, lately has been great because there's a lot of interaction there and connection. So,
Mike, I have a question just to put. I think it could be contextualized, the people that we have here. As you know, we have a lot of PhD students with different backgrounds. We have a real interdisciplinary team. But I would say that we have like three bigger brands of research, which is one of them is like studies in clinical neuroscience and brain health. And we have another big domain, which is like studies in social neuroscience and how some certain social context could modify your biology and how your biology could predict some social behaviors. And I would say we have another different domain, which is more like a specific social decision making in particular social context. We have people working with environment and decision making, but also we have people working on decision making related with arts and another like a ludic scenario. So consider those three big domains like brain health, something more related with social factors and other factors in complex decision making in humans. I was wondering how the epigenetics could be a reliable and available tools in order to see, I don't know, potential biomarkers of certain exposures that we have, or how that kind of measure could be a good tool to track potential changes if you are interested in promoting interventions. And that kind of question is it's just to contextualize for our students how they could approach to this kind of measures. That I think it's a wonderful tool for people working with the interactions between context, exposures, even cultural aspects that could affect your biological system. So
I think in my opinion, the most exciting part is the interventional side.
okay.
It gives you an additional biological measure to show the intervention is changing biology, which potentially could be related to subtle things that maybe clinically yet haven't manifested. So some of these interventions, I'll just give an example, let's just say for cognitive behavioral therapy. If you're looking at a potential outcome measure, maybe on the test you're running in terms of the clinical test or assessment, you don't see a change there at a year, but maybe on some of the biological measures, maybe you see a change in terms of some of the stress genes epigenetically are changing. That may be something that precedes the clinical change at 2 years or 3 years. So it's a way to think of how to follow someone's changing biology that may or may not track at the same time point with the actual clinical investigation. Because the data I was still struck by, right, is I showed you data where we had epigenetics at baseline and we followed cognition and clinical outcomes longitudinally. We never took the epigenetics again in 2 years, but based on where they started at, we could kind of tell which path they went on. So as you kind of are designing longitudinal studies, it'd be nice to have repeated, right, samples and show that the biology is changing. But even at a starting point, we're kind of subgroup stratify. In terms of if you're doing imaging, if you're doing surveys, even the data, I think with, in the HIV context, I didn't get into it, but the stigma aspect there, that's an incredible variable that, if you have an assessment in that regard, that also can show maybe the people that have the biological changes that are most in terms of CD4 counts low, high viral loads, that's due to access to care, but it's also due to stigma. Maybe they feel the strongest stigma. So therefore, right, they're not going into the clinic at least. That's a variable that if you're capturing that in like a clinical way or a survey or a very structural way that you have measurable, quantifiable data, you can substratify groups based on that variable and then see, okay, does the biology also track with it? I think that's the best use of the data in terms of meshing the two. And the nice thing with at least not the single cell data from like lumbar puncture, that's very difficult to do. But in terms of like a blood sample epigenetically, the data is very easy to like access and analyze. So you basically have a spreadsheet with the number from 0 to 100% for each person. And then it's ranked like this. And then it tells you which genes it belongs to. So you can imagine, right? It's a very nice quantitative way to look at associations. But also you can look at group comparisons between disease groups. And that's really depending on kind of like you're saying, those three domains, knowing kind of how people group together. And you guys are probably doing cluster analyses and other things to see maybe in a certain population you're studying, there's like 4 distinct groups. It's very compelling to look biologically. Maybe there's some biology that explains that in terms of how their immune system looks, how maybe, you know, their stress systems look. And that's all based on, I think, when you were born, throughout life, what you've been exposed to, cognitively, how you think of the world, how you change based on, you know, you go to another place, right? I think your worldview changes, you know, all these factors that people are interested in measuring. I think it's a good way to interact the two.
Yeah, for me it was surprise. I mean, it was a big surprise to see that even just some social factors like the social status, or if you experience early adversities, or some measure related to your, if you experience social stress in your life, or if you experience violence or discrimination, even displacement, all of those measures that we could, like a coding, like a social measures and social things, could be tracked by this kind of biological measure. So this, for me, it's a good tool to try to make to create a dialogue between social science and biological science in specific vulnerable populations. This is one of the part of our next grant that we are waiting for new news. I mean, it's like because we are interested in how those social factors could affect the epigenetics, and how those changes in epigenetics could be related with brain health measures in patients with Alzheimer's disease and frontotemporal dementia. But I think that that kind of measure could be a good tool for your projects, because most of you are interested in those kind of complex interactions. Yeah.
I have a question that it might be a kind of follow-up question to that. Is, you mentioned your research has worked with the military at some point. What's their angle? What's their interest in your research? Why?
So in that regard, they're really interested, I think, in pandemic preparedness. So, but with it, the outcome measure of this particular virus, if you're infected with the virus, there's no cure. So it's long-term treatment, but also you see some of the cognitive abnormalities. So it's really, I think, getting a grasp on what's the best way to measure interventions in terms of early treatment. So the data I showed you was this remarkable effort where the US government pumped billions of dollars to give antiretroviral medication to Africa, every single place, to make sure everyone has access so they can't transmit as much. And, you know, we've had, I think, remarkable progress in terms of stemming off some of the untreated people. But as you can imagine, with measuring these social determinants of health factors, it's very difficult to get it to the most needy. Right. They've done a good job, I think, in terms of Asian education, other things with the military. But in this regard, you know, that's really the effort, is a global effort to have access to treatment that's equitable. And for HIV, you know, I think it's been nice to see those relationships were built a long time ago, but it's translated, I think, into why, you know, at the initial phase provided some of the vaccine hesitancy and, you know, a lot of the politicization of things currently with science. That's why we were able to rapidly make vaccines, roll them out. These are things people had at the back end that were really built up some of the HIV field from having kind of, you know, testing, administration of antiretrovirals that were being rapidly developed and other things. But surprisingly, you know, it's been incredible to see kind of their interest too in terms of the mental health aspect, because they see the data and it's the largest driver of health outcomes. So that seems to be a big component to what it is, right? Everything that you guys are studying in terms of the brain, it makes sense, right? You want, for defense or whatever it may be, you want people not to have depression, anxiety, PTSD, you know, these are all things that, but to see potentially maybe environmental exposure could be modifying that, that's worthwhile to study, I think. So that's kind of their angle on it. But it, you know, they also have a lot of, you know, physicians, people, I think, doing a lot of things that, you know, I think are gathering data, which academically has been quite helpful to kind of garner what things look like globally. Because, you know, I think if you look in an area, you'll see a unique context, but sometimes things are shared contextually across places in terms of, you know, stigma. We all feel it in different vulnerable populations, but that general biology potentially of what you feel with stigma may be kind of the same. But, you know, in terms of how that operationalizes throughout different cultures and regions, that would vary. But in terms of like stress systems, and the brain neurotransmission, these are things that I think, it's like the neurotransmitters are different in other regions. So.
Mike, I have a question regarding to one of your slide, which was surprised for me because you have like a bad exposure, stopper mental disease. You have in that imagine like a PTSD, depression, anxiety, like a kind of exposure. I mean, for me, it's understandable if you have there like alcohol consumption, smoking, if you have good physical activity or not, or if you have like a air pollution exposure. But what about the mental disorders considered like an exposure? It's a kind of, because every mental disorder could be, a disease that you could track in terms of epigenetics, which factor affects and which exposure factors interact with genetics for producing or creating a risk for, I don't know, depression, anxiety, and so on and so forth. But what about if you have like a depression? How do you see that affects your epigenetic age?
Yeah, so I think there's two things which we don't have good data to show it's causal. We have consequential data, so it's outcomes. So it could be a consequence of having PTSD or having depression. But surprisingly, if you go just do some Google searching, one of the largest literatures for epigenetics is in PTSD. And I think the reason is because it's such a dramatic change to the biological systems that they're picking up such a strong signal. So that's actually too where the military pumped a ton of funding and data was in that aspect. But in regards to exposure, it seems like it's such a traumatic exposure, but surprisingly too, they don't know at this point, right, data before getting exposed to something in terms of combat or other things that would drive the change. So we don't have longitudinal data to make the link between a cause, but surprisingly, they're interested in studying variability. So this resilience idea we talked about, where you have the same people potentially exposed to the same thing, and for some reason people develop PTSD and some don't. And the thought, at least the hypothesis, is that their starting point in terms of some of the biology is different. So they seem to be more protected in terms of maybe their stress response isn't as great to the stimulus. Maybe something, for example, right? I don't know, the classic example I like to give medical students is if you're a medical student and you haven't seen a cadaver yet, right? That's some people can see the cadaver. Others, like they need to be desensitized to it. So it's just a natural kind of, I think, perception of how the brain is at a starting point that could change kind of through time. But yeah, the PTSD stuff, I've been fascinated too, because it looks like the biology does change with the interventions. Some of the interventions they're doing with desensitization, and things related to PTSD exposure, like some of the ketamine work, it does look like if that tracks clinically, the biology seems to be resetting.
And it looks like if you have like a long disease, mental disease, it's a kind of toxic, like a kind of complicated exposure too.
Yeah, and the thing on the graph I didn't show, which is like, you know this because you see clinically, nobody presents with just one thing, right? Typically, people have lots of health-related issues.
Yeah.
So they're all kind of intertwined and interacting, and this kind of complexity of like figuring out a different context, what's shared and what's not among all different things, because everyone's biology is, for the most part, conserved. We all have the same receptors, genes, pathways, but in terms of the clinical manifestation, each person is so unique in terms of what they are metabolically, mental health, and then it's also very dynamic within a day, a week. So, these are the things that are like these subtle things I think are more difficult to capture, but I think these outcomes that are like ones and zeros, that's where the field has been starting. So it's like PTSD or not, there seems to be some difference there. And then, subtly they bring in like, let's look at depression, no depression. They see 20% is similar between depression and PTSD, but people with PTSD co-present with depression, same as schizophrenia and autism. There's a lot of interconnection there. But yeah, it's I think exciting time right now because I think we have enough data to say it's worthwhile to track this in terms of measure it. The next step is going to be, okay, what do we do with the data as the data just grows and grows and grows? But it'll take, I think, creative people like you guys to access integration of that data with something that's interesting in terms of a measure you're doing for brain health and showing how they can directly connect, because then you can actually see some of the biology that you can target. So
okay, so thank you so much, Mike. Probably students will have time right now or I don't know, more questions, but with the coffee outside of here. Claro, si quieres me llamas, voy a estar libre para una llamadita.